Aerobic respiration occurs within cells. It starts with glycolysis in the cytosol of the cell. It then goes through the link reaction, moving into the matrix of the mitochondria. It then goes through the citric acid cycle within the mitochondria matrix. Then it goes through the electron transport chain and chemiosmosis in the inner mitochondrial membrane, producing energy in the form of ATP. Aerobic respiration is the process in which a glucose molecule is broken down through a series of chemical reactions in the presence of oxygen to release energy in the form of ATP, giving off the products water and carbon dioxide. When oxygen is not present, anaerobic respiration occurs in the form of fermentation or the lactic acid pathway. Aerobic respiration starts with glycolysis. Glycolysis itself is an anaerobic process and can be part of the aerobic or anaerobic pathways. As we move through aerobic respiration, we are going to keep track of how many NADH, FADH2, and ATP are used and produced. In glycolysis, we start in the cytosol of the cell with a single molecule of glucose. There are 10 steps in glycolysis. The first 5 steps of glycolysis are called the investment phase, as they use energy in the form of ATP. The first step in glycolysis involves the phosphorylation of one glucose molecule, which is a 6-carbon molecule, into another 6-carbon molecule, glucose 6-phosphate, using the enzyme hexokinase and the cofactor magnesium. This process takes the phosphate from an ATP and releases a hydrogen molecule. This step uses up an ATP molecule, giving us a net total so far of -1 ATP. The second step involves the conversion of glucose 6-phosphate, a 6-carbon molecule, into yet another 6-carbon molecule, fructose 6-phosphate, using the enzyme phosphoglucose isomerase. This reaction is able to go both ways. The third step involves the phosphorylation of fructose 6-phosphate into another 6-carbon molecule, fructose 1,6-bisphosphate, using the enzyme phosphofructokinase and the cofactor magnesium. This process takes the phosphate from an ATP, creating ADP, and releases a hydrogen molecule. The step uses up a second ATP molecule, giving us a net total so far of -2 ATP. The fourth step involves the breaking of fructose 1,6-bisphosphate into 2 3-carbon molecules, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, using the enzyme aldolase. This is a reversible reaction. The fifth step involves the conversion of dihydroxyacetone phosphate into a second glyceraldehyde 3-phosphate, using the enzyme triose phosphate isomerase. This is also a reversible reaction. This leaves us with 2 glyceraldehyde 3-phosphate molecules that both move through the remainder of the steps. This also ends the investment phase. The next 5 steps of glycolysis are called the payoff phase, as glycolysis starts to produce energy in the form of ATP, as well as NADH, which is used to produce ATP later on in cellular respiration. The sixth step involves the conversion of glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate, another 3-carbon molecule, using the enzyme glyceraldehyde 3-phosphate dehydrogenase and the cofactor magnesium. This process involves using an inorganic phosphate and reducing NAD+ into NADH, as well as releasing another hydrogen. This is a reversible reaction. This step occurs twice, once for each glyceraldehyde 3-phosphate. This step produces an NADH. As it occurs twice, 2 NADH are produced in total for 1 molecule of glucose. So far, we now have a net total of 2 NADH and -2 ATP. The seventh step involves the conversion of 1,3-bisphosphoglycerate into 3-phosphoglycerate using the enzyme phosphoglycerate kinase and the cofactor magnesium. This process involves the phosphorylation of ADP into ATP, taking the phosphate group from 1,3-bisphosphoglycerate and giving it to the ADP. This reaction is also reversible. This step occurs twice, once for each 1,3-bisphosphoglycerate. This step creates an ATP. Since it occurs twice, 2 ATP are produced. We now have a net total of 2 NADH and 0 ATP, as these 2 ATP make up for the 2 ATP used up earlier in glycolysis. The eighth step involves the conversion of 3-phosphoglycerate into 2-phosphoglycerate using the enzyme phosphoglycerate mutase. This process involves shifting the phosphate group from one carbon to another. This reaction is also reversible. The ninth step involves the conversion of 2-phosphoglycerate into phosphoenolpyruvate, or PEP for short, using the enzyme enolase and the cofactor magnesium. This process involves releasing a water molecule each time. This reaction is also reversible. The 10th and final step in glycolysis involves the dephosphorylation of phosphoenolpyruvate into a final 3-carbon pyruvate molecule using the enzyme pyruvate kinase and the cofactor magnesium. This process involves a hydrogen molecule and taking a phosphate group from phosphoenolpyruvate and attaching it to ADP to form ATP. This step creates an ATP. Since it occurs twice, 2 ATP are produced for 1 molecule of glucose. This takes our net total for 1 molecule of glucose to a total of 2 NADH and 2 ATP at the end of glycolysis. The overall reaction of glycolysis is 1 glucose molecule, 2 NAD+, 2 ADPs, and 2 inorganic phosphates gives us 2 pyruvate molecules, 2 NADHs, 2 hydrogens, 2 ATPs, and 2 water molecules. We are now going to look at the second step involved in aerobic respiration, the link reaction. During the link reaction, we will continue to keep track of any NADH, FADH2, or ATP used or produced. In the link reaction, the 2 pyruvate molecules produced in glycolysis are moved from the cytosol via carrier proteins within the mitochondrial membranes into the matrix of the mitochondria. Within the matrix, each 3-carbon pyruvate molecule is converted into 2 2-carbon acetyl-CoA molecules. The enzyme pyruvate decarboxylase removes a carbon dioxide. This step removes a carbon, making it now a 2-carbon molecule. The enzyme pyruvate dehydrogenase reduces NAD+ into NADH plus hydrogen. And coenzyme A also binds to the molecule, creating acetyl-CoA. This process creates 2 NADHs, putting our net total for the link reaction at 2 NADH. These acetyl-CoA molecules are able to directly enter the citric acid cycle and complete the link between glycolysis and the citric acid cycle. The overall equation of the link reaction is 2 pyruvate plus 2 NAD plus 2 coenzyme A gives you 2 acetyl-CoA, 2 NADH plus 2 hydrogen plus 2 carbon dioxide. This takes our net total for 1 molecule of glucose to a total of 4 NADH and 2 ATP at the end of glycolysis and the link reaction. We are now going to look at the third process involved in aerobic respiration, the citric acid cycle, also known as the tricarboxylic acid cycle or TCA cycle. It is also known as the Krebs cycle. At the end of glycolysis and the link reaction, we were left with 2 molecules of acetyl-CoA within the mitochondria matrix that can now enter the citric acid cycle. As we move through the citric acid cycle, we will take note of how many NADH, FADH2, and ATP are used or produced. There are 8 steps in the citric acid cycle. The first step involves the joining of one of the 2-carbon acetyl-CoA molecules with the 4-carbon molecule oxaloacetate, forming the 6-carbon molecule citrate using the enzyme citrate synthase and a water molecule. Coenzyme A is also released. The second step involves the dehydration and then rehydration of citrate into isocitrate using the enzyme aconitase, where a water molecule is released and then a water molecule is added back on. The third step involves the decarboxylation and oxidation of isocitrate, a 6-carbon molecule, into alpha-ketoglutarate, a 5-carbon molecule, using the enzyme isocitrate dehydrogenase. This step involves a carbon dioxide molecule being removed. NAD+ is also reduced to NADH plus hydrogen. This gives us 1 NADH, giving us a net total so far of 1 NADH for 1 acetyl-CoA molecule. The fourth step involves the conversion of alpha-ketoglutarate, a 5-carbon molecule, into succinyl-CoA, a 4-carbon molecule, using the enzyme alpha-ketoglutarate dehydrogenase. This step involves the removal of a carbon dioxide molecule. A coenzyme A group is added, and NAD+ is reduced into NADH and hydrogen. This step creates another NADH, taking our net total to 2 NADH for 1 acetyl-CoA molecule. The fifth step involves the conversion of succinyl-CoA into succinate using the enzyme succinyl-CoA synthetase. In this step, the coenzyme A is removed. It also involves substrate-level phosphorylation of GDP and an inorganic phosphate into GTP. This GTP can then give a phosphate group to ADP to form ATP. This step produces an ATP, taking our net total so far to 2 NADH and 1 ATP for 1 acetyl-CoA molecule. The sixth step involves the oxidation of succinate into fumarate using the enzyme succinate dehydrogenase. This step involves the reduction of FAD into FADH2. This step produces an FADH2, taking our net total so far for 1 acetyl-CoA molecule to 2 NADH, 1 FADH2, and 1 ATP. The seventh step involves the hydration of fumarate into malate using the enzyme fumarase. This step involves the addition of a water molecule. The eighth and final step involves the oxidation of malate into oxaloacetate using the enzyme malate dehydrogenase. This step reduces NAD+ into NADH and hydrogen. This step produces an NADH, taking our net total for 1 acetyl-CoA molecule to 3 NADH, 1 FADH2, and 1 ATP. So far, we have had 1 acetyl-CoA go through the citric acid cycle, but we have 2 acetyl-CoA molecules per glucose molecule, so this cycle would go through twice. This doubles our net total for the citric acid cycle, giving us a final net total of 6 NADH, 2 FADH2, and 2 ATP. This takes our net total for 1 molecule of glucose to a total of 10 NADH, 2 FADH2, and 4 ATP at the end of glycolysis, the link reaction, and the citric acid cycle. The next step in aerobic respiration is the electron transport chain. At the end of the combined steps of glycolysis, the link reaction, and the citric acid cycle, we were left with 10 NADH and 2 FADH2s. During the electron transport chain, these NADHs and FADH2s are used to create a hydrogen concentration gradient between the mitochondria matrix and the intermembrane space. The electron transport chain occurs in the inner mitochondrial membrane. Within the membrane, there are 4 protein complexes involved, complexes 1, 2, 3, and 4. There are also several mobile electron carriers within the membrane: ubiquinone, ubiquinol, and cytochrome C. The electron transport chain starts with complex 1. Complex 1, also known as NADH coenzyme Q reductase, consists of a flavin mononucleotide and a chain of 9 iron-sulfur redox centers. The chain starts with the NADH produced in glycolysis, pyruvate dehydrogenase, and the citric acid cycle, each being oxidized at complex 1 to produce NAD plus hydrogen by the reduction of the flavin mononucleotide FMN into FMNH2. In this process, 2 electrons are transferred from NADH to the reduced flavin mononucleotide FMNH2. These 2 electrons are then transferred to the chain of iron-sulfur redox centers through the oxidation of FMNH2 back into FMN. These 2 electrons then move through complex 1 via the chain of iron-sulfur redox centers from a higher energy state to a lower one. Complex 1 is also a proton pump. Energy is released by the electrons as they move to lower energy states along the chain of iron-sulfur redox centers. This energy is used to pump 4 hydrogen protons across the inner mitochondrial membrane from the matrix into the intermembrane space. This establishes the hydrogen concentration gradient between the intermembrane space and the matrix of the mitochondria. There is a pool of carrier proteins, ubiquinone and the reduced form ubiquinol. At the end of the chain of iron-sulfur centers, the 2 electrons are transferred to the mobile electron carrier ubiquinone one at a time. The first electron is transferred along with the hydrogen, creating the semiquinone intermediate form. The second electron is transferred along with another hydrogen, creating the reduced form ubiquinol. The next part of the electron transport chain occurs at complex 2 and occurs parallel to the processes in complex 1. Complex 2, also known as succinate coenzyme Q reductase or just as succinate dehydrogenase, consists of a flavoprotein, a chain of 3 iron-sulfur redox centers, and a heme B containing subunit. When succinate is oxidized into fumarate in the citric acid cycle, the oxidization produces 2 hydrogens, but it also produces 2 electrons. These 2 electrons are accepted by FAD, which is bound in the flavoprotein of complex 2, reducing it to form FADH2. The FADH2 transfers these 2 electrons to the iron-sulfur redox centers, in turn converting FADH2 back into FAD. The 2 electrons move along the iron-sulfur redox centers until they reach the heme B containing subunit. The heme B containing subunit is the binding site for ubiquinone. It is here that the electrons are transferred to the mobile electron carrier ubiquinone, reducing it to ubiquinol. Complex 2 is not a proton pump. The electrons from FADH2 bypass complex 1 and do not add to the net flow of hydrogen protons leaving the matrix, meaning FADH2 yields less ATP molecules than NADH. From here, the electrons from complex 1 and complex 2 are moved with the mobile electron carrier ubiquinol to complex 3. Complex 3, also known as coenzyme Q cytochrome C reductase, consists of iron protoporphyrin containing cytochrome B, a heme B containing cytochrome C, and one 2-iron 2-sulfur cluster center called the RISC protein. Within complex 3, a process known as the ubiquinone or Q cycle occurs. This cycle starts with one ubiquinol binding with the quinone binding site on cytochrome B called QP. This ubiquinol is oxidized in 2 steps. The first electron is transferred to the RISC protein, which also releases 2 hydrogens into the intermembrane space and converts ubiquinol into the semiquinone anion form. The second electron is transferred to the cytochrome B alheme. This converts the semiquinone into ubiquinone, which leaves the complex to return to the pool of ubiquinone and ubiquinol in the inner mitochondrial membrane. The electron that was transferred to the cytochrome B alheme is then transferred to the cytochrome BH heme. From the cytochrome BH heme, the electron is transferred to the second quinone binding site called QN. At the QN site, the electron converts a ubiquinone into the semiquinone. This semiquinone, in this form, is strongly bound to the QN site until another electron is available to release it in the ubiquinol form. This commences the first half of the Q cycle. The second half requires a second molecule of ubiquinol to be oxidized at the QP quinone binding site. One electron again goes to the RESC protein, again releasing another 2 hydrogen into the intermembrane space. The second goes to the cytochrome BL heme, then onto the cytochrome BH heme. The electron this time binds to the bound semiquinone at the QN quinone binding site, in addition to 2 hydrogens from the matrix, to form ubiquinol that is released back into the inner mitochondrial membrane into the coenzyme Q pool. Complex 3 is a proton pump. It is through this ubiquinone cycle that 4 hydrogens are pumped into the intermembrane space. That concludes the Q cycle portion of what goes on in complex 3. Now we will need to look at what happens to the electrons passed onto the iron-sulfur cluster center of the RESC protein. From the RESC protein, the electrons are transferred to cytochrome C1. From cytochrome C1, the electrons are transferred to the mobile electron carrier cytochrome C. This mobile electron carrier cytochrome C carries the electrons to complex 4. Complex 4, also known as cytochrome C oxidase, consists of a double copper center, a heme site, and a copper plus heme A3 site. Cytochrome C transfers a single electron to the CUA center in complex 4. 4 cytochrome Cs sequentially pass on 4 electrons into complex 4. Each electron is then passed on to the heme A site. From here, the electron is transferred to the copper CUB and heme A3 site. At this site, the 4 electrons split an oxygen molecule into 2 oxygen atoms, which can be denoted as half oxygen. This oxygen molecule is the final acceptor of electrons in the electron transport chain. This oxygen is essential in the electron transport chain as it takes the electrons that would otherwise block the chain up, so it can continue running over and over again. Each half oxygen accepts 2 hydrogen protons from the matrix, each forming a water molecule. Complex 4 is a proton pump. As the electrons move to lower energy states as they move through complex 4, 2 hydrogens are pumped through. This ends the electron transport chain, leaving a high concentration of hydrogen protons in the intermembrane space and a low concentration in the matrix. This concentration gradient is vital for the production of ATP in the next step called chemiosmosis. Chemiosmosis occurs in the inner mitochondrial membrane and is coupled with the electron transport chain. It occurs through an additional membrane protein called ATP synthase, which, as the name suggests, is involved in the production of ATP. ATP synthase is a rotating molecular motor consisting of a F0 subunit, which consists of the rotating rotor that sits in the inner mitochondrial membranes and the stator, which is anchored in the inner mitochondrial membrane. ATP synthase also has a F1 subunit composed of the internal rod that also rotates and the catalytic knobs. At the end of the electron transport chain, we were left with the hydrogen concentration gradient, with the higher gradient in the intermembrane space compared to the mitochondria matrix. The hydrogen protons that are in a higher concentration in the intermembrane space want to move down their concentration gradient into the lower concentrated area of the matrix. These hydrogen protons move through a channel in the stator of the ATP synthase. Once they exit the channel, they bind to binding sites on the rotor. This binding causes a change in the shape of the rotor subunit, which in turn causes the rotor to rotate within the membrane. Once the hydrogen makes a complete rotation, it moves through a second channel in the stator that leads into the matrix. The catalytic knob consists of several subunits, including 3 catalytic subunits that can be found in 3 different states: ADP and phosphate binding state, ATP forming state, and ATP releasing state. The movement of the rotor also causes the internal rod to rotate, which causes these catalytic sites in the knob to activate. The movement of the internal rod causes the shape of the subunit to change, and forces ADP and phosphate already bound in an active site to join to create a new ATP molecule that is bound in the active site. As the internal rod continues to rotate and change the structure of the knob subunits, the ATP is released and is able to bind more ADP and phosphate. Oxidative phosphorylation is the process that occurs on the inner mitochondrial membrane to produce energy in the form of ATP. It involves both the process of the electron transport chain and the oxidation of NADH and FADH to produce a proton concentration gradient, and also chemiosmosis, where the ATP synthase is powered by this gradient to form ATP. Now that we have ATP being produced, what we really want to know is how much ATP is produced in aerobic respiration for each molecule of glucose that initially enters glycolysis. During aerobic respiration, we get 2 ATP directly from glycolysis, 2 from the citric acid cycle. This gives us 4 ATP and a total of 10 NADH and 2 FADH2s that go into the electron transport chain. For each NADH that goes into the electron transport chain, passing on its 2 electrons, 4 hydrogen are pumped in complex 1, 4 hydrogen in complex 3, and 2 in complex 4, giving a total of 10 hydrogen protons pumped for each NADH molecule. For each FADH2 that goes into the electron transport chain, passing on its 2 electrons, 4 hydrogen are pumped through at complex 3 and 2 hydrogen at complex 4, giving a total of 6 hydrogen pumped for each FADH2 molecule. But how many ATP are produced for each NADH and FADH2? This number varies and relates to the number of subunits on the rotor. In each rotation of the rotor on the ATP synthase, 3 ATP are made in the catalytic knob. The amount of hydrogen needed to produce a molecule of ATP depends on the number of subunits on the rotor. This number varies depending on the species the ATP synthase is found in, but usually somewhere between 8 and 15. If it has 9 subunits, it will take 9 hydrogens to rotate once and produce 3 ATP, which equates to 3 hydrogens to make a single ATP. A hydrogen proton is also used in the ATP-ADP translocase to get the ATP out of the matrix, raising this number to 4 hydrogens per ATP molecule. In this scenario, for every NADH with 10 hydrogen protons pumped, it's 2.5 ATP, and for every FADH2 with 6 hydrogens pumped, it's 1.5 ATP. So for 10 molecules of NADH, you would get 25 ATP, and 2 molecules of FADH2 would give you 3 ATP, plus the 2 produced in glycolysis and 2 in the citric acid cycle. You would get a total of 32 ATP per molecule of glucose. Many calculations round up the estimate of ATP to 3 ATP for each NADH, and 2 ATP for each FADH2, which would give a total of 30 ATP from NADH, 4 from FADH2, plus the 2 from glycolysis, and 2 from citric acid cycle, giving a total of 38 ATP for each molecule of glucose. Thank you for watching. If you found this video helpful, be sure to subscribe and hit that like button.